right, welcome to part 3. We were talking about antibodies. Antibodies have basically two roles in B cells. They can act as a receptor for an antigen in the recognition phase of the humoral response, and in the effector phase, they can be produced in large amounts by a clone of B cells. They are then secreted and enter the bloodstream. Once they enter the bloodstream, they then can do two actions. They can bind to the antigen that is expressed on the surface of the pathogen. This stimulates macrophages to ingest or natural killer cells to destroy the pathogen. Action number 2, if the antigen is free in the bloodstream, then antibodies may bind to it to form large insoluble antibody-antigen complexes, which are then ingested and destroyed by phagocytic cells. All right, the last section is going to involve T cells. So T cells, like B cells, also possess specific membrane receptors. You can see that on the picture here. It is not an immunoglobulin. It is a glycoprotein that is made up of 2 polypeptide chains. Each chain, again, has a constant region and a variable region. The variable region provides the site for specific binding to the antigen. There are 2 types of T cells, TH and TC. TH is called a T helper cell. TC is called a cytotoxic T cell. For the TH cell, when they bind to an antigen, it results in the activation of the adaptive immune response. For TC cells, when they bind to the antigen, it results in the death of the cell. Okay. The MHC proteins form complexes with antigens on cell surfaces and assist with the recognition by the T cells, so that the appropriate type of T cell will bind. So these proteins are involved as well with these T cells. There are 2 types of MHC proteins. We mentioned them earlier in the first set of notes. In class 1, MHC proteins, these are found on the surface of every nucleated cell in the mammal body. These type of proteins present antigens to TC cells. These antigens can be fragments of viral proteins or abnormal proteins made by cancer cells. In class 2, they are found on the surface of macrophages, B cells, and dendritic cells, and these present their antigens to the T helper cells. Okay. So this chart was given in your book for table 31.2. It's just telling you that there's any cell, the antigen present as an intracellular protein fragment, and that would be class 1 that's involved, and it would be presented to a cytotoxic T cell, which would kill it. And then if it's a macrophage and B cell, the antigen presented could be a fragment of an extracellular protein. Class 2 will be involved, and that will go to a helper T cell. Okay, so death of target cells. Cytotoxic T cells can produce perforin, which is what is going to kill the bound target cell. TC cells stimulate apoptosis in the target cell, if you remember that. So on your diagram, that picture right there is the infected cell. You can see all those little triangles representing the pathogen. A T cell receptor recognizes the antigen fragment bound to a class 1 MHC protein. This is in your book if you need to go back. T cell releases this perforin, and then the perforin will lyse the infected cell before that virus can multiply. All right, another type of T cell is called Tregs. It's the 3rd class of T cells. Tregs ensures that the immune response does not get out of control, does not spiral out of control. It is similar to the other T cells that we've talked about in that it is made in the thymus gland, it has a T cell and expresses a T cell receptor, and it will become activated if it binds to an antigen MHC complex, as just pictured. However, it is different. It is different than other T cells, and that the antigens they recognize are self-antigens, not non-self-antigens. The activation of Tregs causes them to secrete cytokine interleukin-10, which blocks T cell activation and leads to the apoptosis of the TC and TH cells that are bound to it. The main role of Tregs is to mediate tolerance to self-antigen. Okay, so here is the diagram for Tregs. A T cell, right here, has a T cell receptor for the self-antigen. A self-antigen binds to the MHC and is exposed on the cell surface. You can see that right here. A Treg cell is going to bind to the antigen MHC complex. You can see the binding right there. This binding stimulates Tregs to make the interleukin-10, which suppresses the T helper cells and the T cytotoxic cells and causes it to undergo apoptosis. You can see it disappeared. Right now there are two experimental evidences for this role of the Tregs. They have found that if Tregs are experimentally destroyed during development in the thymus of a mouse, then the mouse will grow up with an out-of-control immune system, or autoimmunity. Another one is called IPEX. It is a rare X-linked or sex-linked disease in infants. It mounts an immune response that will attack the pancreas, it will attack the thyroid, and the intestine. Most affected individuals die within the first few years of life. All right, obviously you can't talk about the immune system without discussing AIDS. For AIDS, you should know what it stands for. It stands for Acquired Immune Deficiency Syndrome. You get AIDS from HIV, which stands for Human Immunodeficiency Virus. AIDS is transmitted from person to person in blood, warm blood, semen, vaginal fluid, or breast milk. No other way you can get HIV. First, it initially infects macrophages. It's attacking the white blood cells. Specifically, it's infecting macrophages, TH cells, and antigen-presenting dendritic cells in the blood and the tissues. So what happens once we have it in there? At first, there's an immune response to the viral infection, and some TH cells are activated. However, because HIV is infecting the TH cells, they are killed both by HIV itself and by the TC cells that lyse the infected TH cells. So, the TH cell numbers decline after the first month or so of contracting HIV. Meanwhile, the humoral immune response is activated due to the extensive production of HIV. It's growing by infected cells. So what's happening is antibodies are binding to HIV and the complexes are being removed by phagocytes. So, because of that, the HIV level in the blood goes down. So now we're kind of in a good spot. We're in a dormant stage. People in this stage carrying HIV feel fine. Their TH cell levels are adequate. They're doing okay. They can mount immune responses against other infections. However, eventually, the virus is destroying the TH cells still, and their numbers fall to the point where they are susceptible to an infection. So eventually a level of TH cells is barely making them. And so now we're getting these infections happening that the TH cells would normally eliminate, but there's not enough of them to eliminate the infection. Infections that they could get now are Kaposi's sarcoma, which is a skin tumor, pneumonia, which is caused by a fungus, and lymphoma tumors, which is caused by Epstein-Barr virus. These infections are called opportunistic infections because the pathogens now are taking advantage of the crippled immune system of the host. Once these infections occur, death is usually within a year or two. And that finishes the podcast and all the notes for Chapter 31.